First of all, I would like to thank the organizer, Carlo, for this invitation. It's a fun session to attend and be part of. So what I'll do today, I will give you an overview of the work that we have been conducting at the Fox Chase Cancer Center here in town on the identification of molecular changes present in single hit cells, cells that have a single mutation in a tumor suppressor gene, and how this may be relevant for cancer prevention. I have no financial relationship to disclose, and I will not discuss any off-label use in my presentation. Let me start by acknowledging many people that were involved in this work over several years, and I would like in particular to highlight a few persons: Al Knudsen, who led this work, and colleagues at Fox Chase, Margie Clapper and Mary Daly, who are at this meeting, Tony Yang, who led the proteomics work I will briefly talk about, Bruce Bowman at Thomas Jefferson University, also in town, and ultimately the big driver of this study, who was and is Levi Kopelovich at NCI, who I also see in the audience. Of course, the premises for the studies are that there is really a compelling need to identify novel, more effective biomarkers for the common carcinomas, breast, ovary, colon, and kidney. And we try to devise a novel system which was different from conventional approaches based on animal models that may not really reproduce well human tumorigenesis, and also different from other approaches based on clinical samples that might be difficult to obtain and invasive for the patients. And so, based on the fact that genomic and proteomic technologies are really ripe for biomarker identification, we devised a genomic, also a proteomic approach to cancer prevention. I will mostly describe microarray aspects of our work, and I'll only briefly touch on proteomics aspects. But the idea is to use, to conduct large-scale gene expression profiling for the identification of very early changes, molecular changes occurring in tumorigenesis. And this work was conducted in the Cancer Prevention Pavilion at Fox Chase that you see in the image. Now, as we all know, the epidemiology of the common epithelial cancers follows this typical pattern in which incidence increases exponentially with time. And this is a reflection of the need for a number of alterations to accumulate in a single cell and its progeny in the transition from a normal epithelium to a benign tumor to cancer. And this is again depicted as a linear model, even though there are, as discussed by Dr. Mali, many problems with this model. But this morphological transition is accompanied by inactivation of tumor suppressor genes such as APC, activation of oncogenes such as KRAS, and it takes time for these events to occur. That, however, is a good thing because we can intervene, and in fact we know that chemoprevention can work, as is the case, for instance, for non-steroidal anti-inflammatory drugs for colon cancer. And in fact, this has been beautifully defined by Al Knudsen in a special issue of Oncology Times that was dedicated to the 25th anniversary of Nixon's war on cancer. If a tumor is caused by a very small number of events, it will be very hard to intervene and prevent it from happening. But if it takes several events, then we will be able to slow the rates at which these events occur, then we may prevent the tumor from arising altogether or to shift the age at which an individual gets cancer by 20 or 25 years, so that people will die for other causes. And so how do we capitalize on multi-step tumorigenesis? The problem of the pitfalls of conventional microarray analysis, where normal tissue is compared to benign lesions and to cancer, is that invariably benign lesions tend to cluster with advanced cancer. And so the critical transition is missed. Now, before an adenoma occurs, which by definition occurs when both copies of the relevant gatekeeper for the tissue tumor suppressor genes are inactivated, there has to be a state in which cells are still morphologically normal, but have an alteration even in a single copy of the tumor suppressor gene. This is what would happen in a sporadic colorectal cancer patient. But in this patient, because these cells are morphologically normal, they will be indistinguishable from normal cells. However, in individuals with predisposition to cancer, in individuals that are carriers for a tumor suppressor gene mutation, such as, for instance, an FAP patient, well, now all the cells of the target tissue, all the cells of the body have a single mutation in the tumor suppressor gene. Then we can identify very early molecular changes by conducting microarray and proteomic comparisons of single-hit cells to normal cells. And in fact, we can imagine that chemopreventive agents may be useful because they may antagonize some of these changes induced by single-hit alterations. Now, we started this work under the aegis of this work statement, with Al Knudson being the PI. And this work statement was entitled Evaluation of in vivo and in vitro pharmacology and toxicology of preventive agents using human mutant cells from dominantly heritable cancers. And the main hypothesis founding this work statement were that these single hit but still morphologically normal, these non-tumor cells from carriers of tumor suppressor gene mutations harbor the earliest and the fewest molecular alterations very early on in tumorigenesis. Again, before tumors even arise. And the idea is that even a small growth advantage could increase the number of one-hit cells may facilitate the conversion to two-hit tumor cells, may facilitate the onset of the benign lesion by which tumorigenesis starts. And the corollary of this hypothesis is that one-hit cells may represent an important target for chemoprevention, as in part alluded by Dr. Malley. Now, are there clues to heterozygous effects? Are there clues to the fact that there are already alterations due to a mutation in a single copy of the tumor suppressor genes? Are there clues that the classical Knudson two-hit hypothesis should actually be modified as to include some very initial alterations present in single-hit cells? There are indeed clues. For instance, clues relevant to colon cancer are listed here. Dr. Kopelovic showed that skin fibroblasts, normal skin fibroblasts from FAP patients have increased sensitivity to transformation by Kirsten virus and by SV40 virus, have altered cytoskeletal actin distribution and increased expression of p53. And Danes show that there is increased detection of tetraploidy in colonic cells from FAP patients. And this is also true for the ovary. For instance, in BRCA2 mutation carriers, the surface of the ovary is not smooth, but has these papillary fissures and deep invaginations. And there are evidences that this is also the case for the breast. Type 1, the morphology of type 1 lobules is altered, as you can see here in normal tissue from BRCA1 mutation carriers. So we conducted this large study by analyzing a number of different genetic conditions predisposing to tumors of the colon, APC mutation, a mismatch repair gene mutation, tumors of breast and ovary, BRCA1 and BRCA2, and tumors of the kidney, mostly tuberous sclerosis and von Hippel-Lindau. Our general approach involved the sampling of normal, the collection of normal tissue, morphologically normal tissue, either from control non-mutation carriers or from tumor suppressor gene carriers, for instance, carriers of BRCA1 and BRCA2 mutations. Typically, we had 6 patients or 6 controls per group. And we obtained primary cultures of epithelial cells from the relevant target organ, or in the case of the colon, cells were derived by laser capture microdissection. So in this case, we have a pathologist look at the LCM material and determine that indeed the crypts that are microdissected here are morphologically normal, whereas in the case of primary epithelial cultures put in culture, we can see that morphologically they are indistinguishable. Let's say BRCA1 or BRCA2 single hit cells are indistinguishable from the control. RNA was extracted, amplified, and submitted to microarray analysis initially with the 40K cDNA arrays and later on Affymetrix arrays. I will now give you examples of our results. Some of these results have been published, and some other results are unpublished and in preparation. So again, for the kidney, we looked at these 2 conditions, tuberous sclerosis complex and von Hippel-Lindau. Those are 2 typical phacomatosis characterized by benign lesions that invariably tend to evolve into cancer. And big clinical problem for both diseases is renal cancer. Now, what we found in our initial study using 40K cDNA arrays is that one finding was that the AKT tuberous sclerosis complex pathway regulates ribosomal protein gene expression. So it is known that the AKT oncoprotein can affect a number of cellular functions, metabolism, survival, proliferation. But the AKT-mTOR axis is involved in regulation of cell growth and protein translation. AKT does that by phosphorylating and inactivating the TSC1 and TSC2 complex. In doing so, it relieves an inhibition on the mTOR kinase. mTOR phosphorylates S6 kinase, which phosphorylates S6, and this leads to an increased translation of ribosomal proteins. And what we found was that, in fact, single-hit renal epithelial cells from tuberous sclerosis patients, and several several mRNAs for ribosomal protein genes had increased expression, suggesting that the control of ribosomal biosynthesis occurs not only at protein level but also at the mRNA level. And this provides even an additional rationale for the use of mTOR inhibitors such as rapamycin or CCI779 for the treatment of colorectal cancer, the treatment of renal cancer. Now, the additional finding that we had concerned hypoxia-inducible factor, or alpha, or HIF. Another consequence of the AKT-mTOR axis is to to increase the expression of HIF, a transcription factor which leads to increased levels of these growth factors listed here, which are important for tumor growth and angiogenesis. Now, normally, the VHL oncoprotein suppresses HIF. And we found that in cells with single inactivation, as would be expected by the known biology of the AKT-mTOR HIF pathway, in cells with single hit inactivation of TSC1 or 2, the levels of the HIF mRNA were increased. And this, again, provides an additional validation for the use of HIF-1 alpha inhibitors for renal cancer. Now, we are conducting now additional studies using the Affymetrix platform, which is more precise and more sensitive. And this is a study that is almost ready to be submitted. And we find, again, in single-hit VHL TSC renal epithelial cells, gene expression differences compared to wild-type renal epithelial cells. There are about 1,300 genes for VHL and 70 genes for TSC, which discriminate between VHL single-hit cells and wild-type cells, or TSC single-hit cells and wild-type cells, respectively. A gene ontology analysis of these genes reveal several functional categories that are listed here. In red, we have functions, biological processes enriched in upregulated genes. In green, enriched in downregulated genes. And there are differences in response to oxidative stress, in aerobic respiration in the case of VHL cells, which led us to uncover an interesting connection between hypoxia and the Krebs cycle that will be described in more detail in the paper. And similar analysis in the case of TSC single-hit cells revealed the connection between Wnt signaling and the AKT-mTOR pathway. Now, moving on now to the breast, again, we looked at BRCA1 and BRCA2 mutation carriers. And in work that we published this year, we identified gene-specific single-hit, gene-specific and tissue-specific single-hit cells. For instance, this table lists alterations in the level of mRNAs in BRCA1, specific for BRCA1 or BRCA2 single-hit cells for the breast. And as you can see, there are a number of these genes which were validated this is the fold change detected with the Affymetrix platform, and these changes were validated by a microfluidics real-time PCR approach. And I would like to point your attention to some secretoglobin genes. Those are mammaglobin and lipofilin B. These genes are upregulated in BRCA1 single hit cells, and very interestingly, both mammaglobin and lipofilin B had been proposed as biomarkers for breast cancer. Another interesting potential biomarker is chitinase 3 like 1, a gene that has been proposed as a biomarker for glioblastoma. Now, these changes are also tissue specific. So here we have differences in single hit cells from ovarian, single hit epithelial cells from the ovary. And here I would like to point your attention to serum amyloid A1 and A2 mRNAs. And interestingly, these serum proteins had been previously proposed as biomarkers for ovarian cancer. Finally, I would like to give a brief description of some of the functional significance of these altered gene expression patterns that we have detected. And in the case of the BRCA1 single-hit breast epithelial cells, again, these functional aspects are consistent with the known biology of BRCA1. So, for instance, a specific bioinformatic analysis called the GSEA allowed us to detect gene expression signatures enriched in breast BRCA1 single-hit cells that are similar to those detected in stem and progenitor cells, which is consistent with the proposed role of BRCA1 in regulating stemness and differentiation of breast progenitor cells. And is consistent also with two studies that are kind of similar to ours. The enrichment in luminal progenitor cells in breast tissue from BRCA1 mutation carriers, and in another microarray-based study, the enrichment of stem cell genes in breast tissue from, again, from BRCA1 mutation carriers. Now, similarly, single hit changes have been detected and are being detected in the colon. In this published study conducted by with a proteomic approach, crypts were isolated from APC mutation carriers from FAP, familial polyposis patients, and controls, and their proteomic profile was analyzed. About almost 1,700 proteins were detected with a 2D gel approach followed by mass spec identification. And in this analysis, more than 10%, 13% of the proteins were differentially expressed between FAP single-hit cells, morphologically normal cells, and controls. This is one example. This is a superoxide dismutase 2 gene, which has a higher expression with detection of higher number of isoforms in FAP single-hit crypt cells compared to controls, and all the altered processes regulated by the differentially expressed proteins are again consistent with the APC function: apoptosis, cell adhesion, motility, cytoskeletal organization, mitosis, and oxidative stress. And we are finishing up a similar analysis conducted this time with a microarray approach on laser capture microdissected colonic crypt cells from FAP patients and controls. And again, we find hits on signal transduction, fatty acid metabolism, carbohydrate metabolism that are consistent with the function of of APC. So let me summarize by saying that hereditary cancer syndromes provide a unique opportunity to develop and test novel prevention strategies. When highly sensitive techniques such as microarray analysis and proteomic analysis are used, heterozygosity for tumor suppressor genes is associated with detectable changes in the expression profile, and these changes may represent the earliest molecular changes in tumorigenesis. Some of those are markers of transformations, and others are drivers of the transformation process, and such are potential targets for chemoprevention. In most cases, the changes detected are consistent with the known biology of the relevant tumor suppressor gene for the target tissue involved. And as I showed you for the example for the kidney with the mTOR and IF1 alpha inhibitors, these genomic and proteomic-based approaches may allow the identification of druggable targets. So in the future, we are collecting additional data, performing validation studies. The idea is to try and identify both tissue-specific as well as general molecular alterations, and link them to an increased risk of cancer, something that formally we have not done. This has obvious translational implications, both in terms of diagnosis, for instance, development of tests to identify heterozygosity for mutant tumor suppressor genes and stratify the population into classes of cancer risk, and in terms of therapeutic applications, such as development of targeted therapies with novel chemopreventive agents. And here one implication is that because we see the involvement of classical signaling pathways in single-hit cells, perhaps these chemopreventive efforts could piggyback on cancer therapy efforts. And in fact, since tumor suppressor gene mutation carriers may benefit greatly from aggressive chemopreventive strategies, for those patients, one can even allow a degree of toxicity that will not be suitable for chemopreventive approaches for the general population. And I'll stop here and thank you for the attention.
Questions?
So if some of these genes are to be targets, then of course you want to know if they're causal, and maybe even you want to know if they do more than correlate with increased risk. Can you do some experiments to introduce those genes into the one-hit mutation cells and see if you can induce progression by some measure of further advanced phenotype? I note that one that you found, osteopontin, is known to be a driver of progression, both tumor promotion and tumor progression. So, but I wonder, I guess a lot of them that you found, you just don't know. And I wonder if you're trying to reconstitute that in vitro.
Yes, so we can do, those are very good points. We can do some of these experiments, and we are trying to start them. The difficulty here is that we are dealing with not established cancer cell lines. We are dealing with early passage strains. We shouldn't call them lines, with limited lifespan. So that's the limitation. But certainly we could try and perturbate the pathway and follow the output, for instance, by gene expression changes, where we don't need a large number of cells to...
Or too many doublings.
Or too many doublings, correct.
Yeah.
Correct.
But maybe one of the endpoints could even be extended lifespan.
Yes, yes. In fact, we have another paper that hopefully will be submitting soon, where we find that we can immortalize, with certain conditions, colonic epithelial cells but only if they are from FAP patients. As you probably know, colonic epithelial cells, when put in culture, die shortly thereafter. But yes, under certain conditions, they can be long-term.
I'm actually interested in the hypothesis 2 of the work program, which is that a small increase in fitness in either proliferation or survival can have a dramatic effect on progression. We certainly see this in our models that if the first hit in tumor suppressor gene really is a recessive effect, and so it's a neutral mutation, you get very little progression, and it's a real bottleneck to progression. But if there is some kind of fitness effect on that first hit, like you're seeing these at least phenotypic effects, the question is it actually changing the fitness? And so how are you going to assay the change, the relative increased fitness of these single hits?
I guess one approach could be to again, with the limitations of having to deal with the strains, not with the established lines, would be to mix single hit cells with plus cells in culture and see if there is a growth advantage that can be detected within a few passages that we can work with. So that might be an approach.
Good. All right. Well, let's thank Alfonso once again, and we'll have Dr. Pepper up to the stage.